Good morning, good afternoon, and good evening to everyone joining us from wherever you are around the world. Distinguished guests, ladies and gentlemen, it is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with our main presentation, allow me to provide a brief overview of the initiative that brings us together today. Install Africa, Innovation, Science and Technology for Action and Learning in Africa is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Install Africa was born from a collaboration between the Foresight Institute of Research and Translation Rwanda, or FIRAT Rwanda, and its technical partner, DROS Africa UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, it is now my distinct pleasure to introduce our invited guest speaker.
Ladies and gentlemen, it is our great pleasure to introduce our distinguished guest speaker for the 21st Install Africa webinar, Dr. Neza Buhadu. Dr. Buhadu is an associate professor of physiology and neuroscience at the Faculty of Sciences, Mohammed V University in Rabat, Morocco. She is a dedicated researcher in African neuroscience with a strong focus on understanding the biological mechanisms of neurodegeneration and exploring innovative neuroprotective strategies. As a member of the Physiology and Physiopathology research team, her work examines the impact of environmental neurotoxicity on brain health. Using experimental models, she investigates how oxidative stress contributes to the onset of Parkinson's disease, while also exploring the potential of natural neuroprotective agents, including Moroccan medicinal plants, as well as the role of physical activity in preserving dopaminergic function. Her research has been published in leading journals such as ACS Chemical Neuroscience and Frontiers in Neuroscience. In 2024-2025, Dr. Buhadu was awarded the prestigious NIDA Invest Fellowship, which supported a research visit to Dr. Peter Calivas's laboratory at the Medical University of South Carolina. This experience enabled her to explore the neuroplasticity of stress response circuits, expand her research expertise, and strengthen scientific collaboration between Morocco and the United States. Beyond her research, Dr. Buhadu is deeply committed to scientific capacity building across Africa. Since 2017, she has served as the coordinator of the IBRO UM5 Center, contributing to the training of over 360 young African researchers through intensive thematic schools. She has also served on the executive committee of the Society of Neuroscientists of Africa and has been actively involved with the International Society of Neurochemistry, reflecting her dedication to advancing the global scientific community. As a speaker for Install Africa, Dr. Buhadu brings a valuable perspective on the role of fundamental research in advancing our understanding of environmental health and developing therapies for neurodegenerative diseases. Dr. Buhadu, we are honored to have you with us today. The floor is yours.
So first thing, good morning, good afternoon, everybody. Thank you for joining us today. I would like to begin by thanking FIRAT Rwanda and the Install Africa webinar series for their invitation and the organizing committee team for bringing together African and international neuroscientists in this exchange. So the title of my presentation today is From Experimental Models to Therapeutic Strategies for Parkinson's Disease, Environmental Neurotoxicity and Neuroprotection. So over the next 30 or 40 minutes, I will outline how our laboratory uses environmentally relevant rodent paradigms to dissect dopaminergic vulnerability, and how this experimental framework underpins our work on neuroprotection, including a chronotherapeutic dimension that defines the next phase of our work. So my presentation is outlined, or the lecture is organized in 3 parts. The first part sets out the rationale for environmental modeling and develops the convergent pathophysiological cascade. The second part examines our work on neuroprotection, phytochemical, behavioral, and computational analysis. The third part, or part 3, introduces the chronobiological dimension and our future projects. I will close with open scientific question and an invitation to collaborate. So why PD, or Parkinson's disease, needs better models? Let me begin with the scale of the clinical problem. Approximately 10 million people currently live with Parkinson's disease worldwide, and the prevalence is projected to double by 2040. Parkinson's disease is now the fastest growing neurodegenerative disorders in our century. Despite this trajectory, the therapeutic landscape has changed remarkably, has little over recent decade. L-DOPA, dopamine agonists, monoamine oxidase B and COMT inhibitors all relieve motor symptoms, but none of this modify disease progression. No disease-modifying therapy is yet approved. A second observation on this slide is essential to our framework. The identified Parkinson's disease genes like SNCA, PINK1, or Parkin together account for fewer than 10% of cases. The remaining 90% are sporadic and multifactorial, driven by aging, environment, and gene-environment interaction. This distribution defines the central premise of our research: to evaluate disease-modifying strategy in a way that is relevant to most patients. We need an experimental system with construct, phase, and predictive validity for sporadic PD, and environmental neurotoxin paradigm meet this criteria. So the epidemiological relevance supporting environmental contribution is robust and reproducible. Across more than 50 concordant case control studies published since 1995, occupational and environmental pesticide exposure is associated with a 1.5 to 2-fold increase in PD risk. This signal is consistent across continents and is reinforced by association with rural residents, well water consumption, and farming exposure. 3 classes of exposure occur in the literature with sufficient frequency to merit experimental property. First, pesticides, paraquat, maneb, rotenone, etc. Second, heavy metals like manganese and lead. And third, air pollutants like fine particulate matters and traffic-related particles. This exposure share 3 properties of methodological value. They are environmentally relevant, mechanistically tractable, and reproduce Parkinsonian pathology in rodent models. They therefore offer collaborable entry point to integrate causal and test neuroprotective interventions. Regarding pathophysiological cascade, although this exposure differ widely in their primary chemistry, they converge on a shared molecular cascade, and this convergence is what makes mechanism-targeted neuroprotection feasible. The cascade begins with oxidative stress and mitochondrial dysfunction. Reactive oxygen species accumulate, and complex 1 and complex 3 on the respiratory chain are inhibited. This translates into measurable damage markers, depletion of superoxidase SOD and catalase activity, and accumulation of malondialdehyde. These are precisely the markers we quantify in our experimental tissue. Downstream of the oxidative stress burden, alpha-synuclein undergoes misfolding and aggregation, producing Lewy-like pathology and proteostatic failure. The pathology is amplified by neuroinflammation, microglial activation, and pro-inflammatory cytokine release. The convergent endpoint is dopaminergic neuron dysfunction and loss in the substantia nigra pars compacta, which reduced striatal dopamine release. The clinical correlate is the Parkinsonian phenotype with motor signs such as bradykinesia and rigidity, together with the non-motor manifestation as cognitive affects and autonomic disturbances. Environmental exposure are heterogeneous in their starting chemistry, but they engage a shared molecular cascade, and it's this convergence that licenses a unified experimental and therapeutic framework. So this brings me to the framework that organizes our work. The central question displayed on this slide is: how can environmentally relevant animal models inform mechanism-based neuroprotective strategies in PD, in Parkinson's disease? We address this question along 3 integrated axes. The first one is the environmental neurotoxicity by reproducible rodent model of PD using either lead, manganese, and paraquat maneb to dissect mechanism of dopaminergic vulnerability. The second axis is the neuroprotective strategies, pharmacological and non-pharmacological interventions like phytochemicals, melatonin, and exercise were validated within this toxicant model. And the third and final axis is the chronobiology of melatonin, since the circadian regulation of dopaminergic excitability and neuronal vulnerability with a view toward chronotherapeutic strategies. This structure of the lecture follows these 3 axes. So let's start with the part 1, which is the models of environmental Parkinsonism. Let me now turn, we work with this complementary toxicant paradigm in male Western rats, mainly. I also use female Western rats, but I just started using females. All my results presented today are from male rats. So started with lead exposure administered chronically through intraperitoneal injection, and we targeted. So the lead is targets the hippocampal and nigrostriatal circuits and supports both developmental and adult onset paradigm. Second model I used is the manganese. Given chronically by intraperitoneal injection, it accumulates selectively in the basal ganglia and reproduces manganese, the Parkinsonian syndrome described in occupational exposure. And the third model is the paraquat maneb, which is our most extensively characterized model. We administered paraquat at a dose of 5 milligram per kilogram, combined with maneb at a dose of 0.05 milligram per kilogram intraperitoneally twice weekly for 6 weeks. The combination produces synergetic mitochondrial and alpha-synuclein toxicity. Across all these three paradigms, we apply a multi-scale analysis framework, starting from behavioral tests, batteries like Rotarod, beam walking tests, elevated plasmase, etc., biochemistry analysis by analyzing the melandiolide levels, the SOD catalase in the prefrontal cortex, striatum, and hippocampus. And we also did an in silico study with molecular docking to evaluate the alpha-synuclein interactome, specifically targeting melatonin, how melatonin and if melatonin interacts directly with alpha-synuclein and prevents its aggregation. So this slide represents the behavioral signature of the paraquat and maneb model. This work was published last year in neurochemical research with my doctoral student Yusuf Mkhlov as first author. Three behavioral domains are productively. So let me first explain how we did. So here you can see the animal was injected with paraquat and maneb for 6 weeks, as I said before, like it's twice a week intraperitoneally with the combination of paraquat and maneb. And after the 6 week, the treated animals received L-DOPA as the reference treatment for Parkinson's disease in humans. So we have a control group who received just the vehicle solution, which is sodium chloride, for 6 weeks, and then continued the vehicle solution for the remaining 3 weeks or treatment phase, which is the 3 weeks. Then we have the second group, which is the paraquat and maneb intoxicated animal who received the combination of paraquat and maneb for 6 weeks and no treatment after. And our third group, which is the intoxicated with the treatment, L-DOPA treatment. So these animals received 6 weeks of paraquat and maneb combination, and at the end of the inductive phase, they received L-DOPA and a combination of L-DOPA and carbidopa for 3 weeks. At the end of the treatment phase, all animals underwent an assessment phase with behavioral tests and biochemical analysis. So as you can see in the tables here, I'm just focusing on the red underlined graphicals. So for behavioral tests, which is this part of the slide, so you can see for the APM, for example, we have the control at the left side, and here we have the intoxicated and intoxicated with treatment. So we can see in the motor domain, animals show decreased rotarod latency here. Sorry, and impaired beam walking performance, which is a clear bradykinetic profile. In affective domain for the EPM, elevated plasmase, paraquat and maneb increases time spent in the closed arm, so time in the closed arm of the elevated plasmase, and increases immobility in the forced swimming test here. Robust readout of anxiety and depressive-like behavior. In the cognitive domain, we have the novel object recognition. Recognition memory is impaired as indexed by reduced object recognition discrimination. So this is what face validity looks like in practice. The model recapitulates the constellation of motor and non-motor manifestations that define clinical Parkinson disease. And we can see that levodopa, L-dopa, improves cognitive, here. So improves cognitive, significantly improve cognitive symptoms, in novel object recognition, we have no significant effects. In forced swimming tests, which is depression, we have a significant effect. And in motor tests like Rotarod, we have a significant improvement of levodopa treatment in paraquat and maneb animals. Regarding the biochemical analysis, I showed here just the melandialdehyde levels in prefrontal cortex and striatum. Again, here we can see that compared to the control, the paraquat and maneb animals have more melandialdehyde, meaning that we have an oxidative stress and in process, and the levodopa treatment enhances this condition by reducing the melandialdehyde expression both in prefrontal and striatum levels. I'm sorry for my English is not my first language, so sometimes I need to look forward to express myself. But I hope it's not a problem, and you can still understand what I'm trying to say. Concerning SOD, superoxide dismutase, same profile. So compared to the control, paraquat and maneb animals express less superoxide dismutase, meaning that the antioxidant defense is impaired, and levodopa has a significant effect on improving the expression of SOD. So here I'm building on this validated model to expose the part 2, which examines the neuroprotective strategy. We pursue both pharmacological and non-pharmacological approaches. The pharmacological arm comprises 3 lines of investigation: phytochemicals, Manta rotontifelia aqueous extract with lavandula species in progress. It's a work that we are still doing. We also explored the endogenous chronoprotective molecules like melatonin, both as antioxidant as circadian signal, and we did an in silico exploration with narangenin as a candidate alpha-synuclein binder. The non-pharmacological arm centers on moderate physical activity, applied both therapeutically following toxicant exposure and as a pre-conditioning intervention. I will present each line in turn. Okay, so concerning the Manta rotontifelia work published in Frontiers in Neuroscience in 2023 with Khadija Boualem, which was a PhD student at that moment in our lab. The induction model we just employed hydrogen peroxide as a oxidative stress model. It was like a first work to explore how it goes with what we had as equipment, as material at that time, before going to using the paraquat and maneb. So we used 0.5% hydrogen peroxide in drinking water for 14 days, which is a subchronic in vivo oxidative stress challenge. Animals receive around the plant or the extract as co-treatment. The phenotypic characterization cover three layers again. The behavioral battery with the open field, rotary, Y maze, cell immersion, etc. The hepatic biomarker with ELT, AST, and bilirubin are quantified to access systemic protection, and the extract itself is profiled by LC-MS MS profiling. Phytochemical analysis identifies four major phenolic components with the strong DPPH and EBATase radical scavenging activity, and the half effective concentration are around 26 and 21 microgram per milliliter, comparable to reference antioxidants. The in vivo results displayed on this slide demonstrate three principal effects. So the aqueous extract reversed the H2O2 induced motor and cognitive deficit recovery in rotary, nociceptive threshold, and open field activity. You can see here for rotary, for example, we had administered three different doses. So compared to the control, in H2O2 animals, we have an increased time spent in rotary compared to the control. Same for pain with the tail immersion test and with activity in the open field. Montarodontifelia, it protects also the hepatic biomarker with significant reduction in ALT and EST. You can see it in the table here. So if you, for saline group, we have ELT levels, and when we administer doses of our plants, we can see that it's proportional to the concentration. We have a reduction in ELT or EST levels, meaning that it has a beneficial effect on hepatic biomarkers, and it restores also the antioxidant balance. So the translational interpretation requires care though. This finding constitutes mechanistically suggestive evidence that ethnopharmacology-guided discovery can yield neuroprotective candidates. They do not yet constitute disease-modifying evidence, but they establish a credible pipeline for further validation. Okay, so concerning exercise as non-pharmacological intervention, our working hypothesis is that moderate structured exercise engages endogenous antioxidant defense and dopaminergic circuit plasticity, attenuating both motor and non-motor manifestation of environmentally induced Parkinsonism. The protocol was structured as follows. So paraquat and maneb induction is administered over 6 weeks. So we have same concentration as described before, 5 milligrams per kilogram of paraquat combined with 0.05 milligram per kilogram of maneb. So administered twice a week for 6 weeks. Animals then undergo 4 weeks of progressive treadmill training. So I'm showing here in this table the process or the protocol for exercise or rotary exercise training. So 30 minutes per session, so 3 sessions per week, with speed escalating from 10 to 20 RPM by the end of the study. A subset receive concurrent L-DOPA, carbidopa, modeling clinically realistic adjuvant therapy, behavioral, biochemical, and histological assessment flows. So we had the induction phase with paraquat and maneb or toxicant injection, concomitant with rotarod training at low speeds, constant speed of 8 rotation per minute. And then by the end of the induction phase, we move to the treatment phase. So we are administering exercise as a treatment therapy. For 3 weeks. Animals undergo physical training with an increasing speed of the rotarod for 3 weeks, and then we go for the behavioral tests and after sacrifice for the biochemical analysis. So the data demonstrates convergent rescue across 3 behavioral and biochemical domains. Motor function recovers. So you can see here the group with paraquat maneb and exercise, and paraquat maneb exercise with levodopa. On the top part, we have all behavioral tests here. And in the right side, we have the biochemical results. So anxiety-like behavior here in the EPM or elevated plasmase, indexed by elevated plasmase matrix, is attenuated, and critically antioxidant enzyme activity like superoxide dismutase here and catalase is restored toward control values in prefrontal cortex and striatum. Of particular translational interest, the combination with levodopa further improves motor recovery without compromising the antioxidant rescue. This positions exercise not as an alternative but as a mechanism grounded adjuvant to dopaminergic pharmacology. We suggest that exercise should be introduced as a complement to the pharmacological treatment. We have extended this work into a preventive paradigm. So this work is submitted, the paper is submitted, and we are waiting for reviewers' comments. So we did the training or the physical training before the exposition to the toxicant, to the paraquat and maneb, again for 3 weeks. So yeah, the research question here is: does physical exercise preconditioning confer neuroprotection against paraquat maneb induced Parkinsonism both before and during the exposure? So this study, currently under review, as I said, employs treadmill training initiated before and continued during the toxicant exposure window compared with sedentary cohort. The data displayed indicates that preconditioned animals show a substantial prevention of motor function evident in rotarod and beam walking performance. So agricultural workers and other groups exposed to pesticide for whom structured physical activity could be evaluated prospectively as a preventive strategy. What about pharmacological neuroprotective using melatonin? So I now introduce melatonin, which provides the conceptual bridge into part 3 of my lecture. In a manganese-induced Parkinsonism model conducted with my master's students, Jihan and Habiba, we evaluated exogenous melatonin at a dose of 20 milligram per kilogram, administered alongside with manganese insult or manganese toxicity. So the findings are encouraging, even if we didn't have enough animals in our cohorts, and we need to develop more this work to have more significatively or more at least effective animal number to have significant results. And so melatonin restores rotarod. Here we have manganese, melatonin, and melatonin plus manganese group versus control. So we did rotarod, sucrose preference test, open field test, and forced swimming test. And we only did malondialdehyde level, MTA levels in the frontal cortex and striatum. So we show these results show that melatonin restores rotarod performance, rescues sucrose preference as an index of anhedonia, reduces forced swimming test immobility, and attenuates anxiety-like behavior in the open field. MTA levels in frontal cortex and striatum trend toward control values. So this data illustrates the cytoprotective dimension of melatonin, antioxidant action, mitochondrial protection, and behavioral rescue, as described in the literature. Melatonin, however, presents a second and opposing pharmaceutical dimension that I will develop shortly. So we move on with Habiba's work to the in silico targeting alpha-synuclein, because when we had this like controversy between behavior and biochemical results, we asked ourselves the question: is melatonin acting by like a scavenger by linking to the alpha-synuclein and preventing alpha-synuclein from aggregation? So we tried with in silico workflow to determine if melatonin link or can be recognized by alpha-synuclein and aggregate to alpha-synuclein. So Habiba did the work and found that melatonin doesn't have any possibility to link with the alpha-synuclein. But when doing this work, she screened phytochemicals from ethnobotanical sources through ADMET filtering, followed by molecular docking against monomer alpha-synuclein using Autodoc Vina. Top scoring component undergo stability assessment and are advanced in to in vitro and in vivo validation. So our leading candidate here is the narangenin, flavonoid of citrus origin. It shows stable interaction with the alpha-synuclein binding pocket, a favorite ADMET profile, including predicted blood-brain barrier permeability and low predictive toxicity. The anti-aggregation rationale is supported computationally by cellular validation is underway, and the manuscript is in preparation with Miss Habiba Taher. So we move on to the chronobiology of the dopaminergic system. I now enter the third part of my work, which represent the principal direction of my future research. So the dopaminergic system is fundamentally rhythmic. Pineal melatonin is synthesized under suprachiasmatic control and peaks during the dark phase, as we all know. And striatal and retinal dopamine show the opposite phase. So dopamine peaks during the active phase, melatonin peaks during the inactive phase. So the two systems are organized in an anti-phase manner. They are also reciprocally coupled. Melatonin modulates dopaminergic synthesis, release, and receptor expression. including direct effects on tyrosine hydroxylase. Conversely, dopamine regulates the local clock. Nigrostriatal dopaminergic input is required for normal PER2, which is period 2, which is one of many clock genes that regulate rhythmicity in cells. So the nigrostriatal dopaminergic input is required for the normal rhythmicity of PER2 in the dorsal striatum. So 3 implications for Parkinson's disease flow. The first one is dopaminergic loss blends striatal period 2 rhythmicity or rhythms, contributing to the sleep, mood, and autonomic non-motor symptoms patients describe, which appear way before the motor symptoms. Second application is the relationship, since it's bidirectional, circadian disturbance itself amplify oxidative stress, mitochondrial dysfunction, alpha-synuclein aggregation, and dopaminergic vulnerability. And the dopamine, again, as I mentioned here, it disturbs period gene or rhythmicity. Third implication or third part, and this is the conceptual basis of chronotherapy. This bidirectional coupling provides the rationale for time-targeted intervention, timed light exposure, timed melatonin administration, sleep-wake stabilization, and time-optimized dopaminergic medication. So our experimental approach is summarized in this slide. The working hypothesis is direct. Pineal melatonin is a chronoprotective antioxidant, and disruption of melatonergic signaling increases dopaminergic vulnerability to oxidative stress. We address this hypothesis through 2 complementary entry points. The pharmacological one, already presented, uses exogenous melatonin in the manganese paradigm, and the endogenous one uses genetic dissection by comparing 2 strains of mice, C57B, which are constitutively melatonin deficient, with CBA mice, which retain melatonin synthesis, combined with surgical isolation of the pineal out or the ganglia output by superior cervical ganglionectomy in rats. So I now arrive to the central paradox that defines this field. Melatonin exhibits 2 opposing actions in the Parkinsonian brain, which were described in the literature. The cytoprotective action compromises direct free radical scavenging, antioxidant signaling, anti-inflammatory effects, and mitochondrial protection. In PD models, melatonin preserves substantia nigra dopaminergic neurons and improves motor and cognitive performance. The anti-dopaminergic action consists of direct inhibition of striatal dopamine release. Melatonin acts via MT1 and MT2 receptors coupled to GIO signaling and may exacerbate motor symptoms when administered in inappropriate circadian phases. So this paradox has constrained the clinical translation of melatonin in Parkinson's disease. It also represents a precise scientific opportunity, or our hypothesis is that the cytoprotective and antidopaminergic actions of melatonin are temporally and pharmacologically dissociable, that they operate through distinct signaling pathways with distinct kinetics, and that the dissociation can be explored to define chronotherapeutic windows in which the protective action prevails. So this hypothesis is operational in our MEL Park, what I call MEL Park projects, structured around three nested questions. First question, the mechanism. How do MT1 and MT2 receptors integrate with D2 auto receptor signaling at striatal dopaminergic terminal across the circadian cycle? And can the cytoprotective and antidopaminergic action of melatonin be functionally separated through receptor biased ligands? Question two is the vulnerability. Does dopaminergic susceptibility to environmental neurotoxin follow circadian rhythms in vivo? Can chronotyping inform individualized risk satisfaction in human PD cohort? And the third question is the translation. Can chronotherapeutic delivery of melatonin or agonists modify the disease trajectory? What peripheral biomarkers, for example, BDNF in platelets, or sleep architecture, best track central response? The multi-method strategy addresses this question through six complementary approaches: mouse model with dissectable melatoninergic signaling, striatal slice, first scan cyclic voltammetry at defined circadian time points, for example, ZT, Zeitgeber time 10 or 22, which are the times of the peak and the nadir of melatonin expression or presence, signaling, which with or without neurotoxin exposure, patch clamp recording on identified dopamine neurons, chemogenetic gain or loss of function manipulation of MT1 and MT2 signaling, behavioral phenotyping, cross-motor and non-motor domains, and single-cell transcriptomics on midbrain dopamine neurons. Environmental modeling, multi-scale mechanistic readouts, and neuroprotective intervention into a single chronological project. So our work is actively open to collaboration along this direction. First, models and methodology. So we are open to Drosophila and zebrafish Parkinsonian models, mid-brain organoid, and advanced electrophysiology and imaging platform. Second, therapeutic angles, phytochemistry, exercise, and rehabilitation paradigm, melatoninergic ligands, melatonin receptor agonists, and integrative omics. And third, the translation, environmental exposure cohorts, peripheral biomarker, including platelets, BDNF, clinical PD networks, and chronotyping protocols. Investigators with interest in any of this direction are warmly invited to contact me. My coordinates appear on the slide, on this slide, and on the closing slide. So the take-home messages as a conclusion: first, environmental neurotoxicity provides a tractable model of Parkinson's disease. Pesticide and heavy metals reproduce the motor, non-motor, and oxidative stress signatures of clinical PD in robust rodent paradigm, establishing a rigorous platform for mechanistic and therapeutic investigation. Second, neuroprotection in multimodal, pharmacological, and behavioral in nature. Phytochemicals, exercise, and melatonin converge on shared antioxidant and dopaminergic mechanisms. Combining and adjuvant approaches deserve systemic evaluation alongside dopaminergic pharmacotherapy. And third, chronobiology constitutes a frontier for Parkinson's disease therapeutics, resolving when melatonin protects, not only whether it does, may unlock a chronotherapeutic class of intervention and refine our understanding of dopamine vulnerability. So I would like to acknowledge that this body of work is collective. I'm grateful to my team, physiology and physiopathology research team at the Faculty of Science, my mentor, Professor Nouria Khadr Ghazal, Professor Peter Calavas, and all my students, Siham, Jihan, Khadija, Habiba, Nora, Rita, my friends and collaborators, Insaf in University of Alabama, Omar in Dublin, Royal College of Surgeons in Dublin, Hassan in Morocco, here in University Mohammed VI, and Yasin from the same university, University Mohammed V. Thank you for your attention and welcome your question. So, yeah, I see the first question. Are there critical exposure windows, for example, early life versus adulthood, that matter most? Yes, but I didn't do any experiments on different ages. We mainly use adult animals, but I can see for say, for example, that even when we did the male female experiment, we had different outcome in terms of behavior or biochemical effects. So I guess early life or adulting, very aged animals will show different outcomes. But I didn't do the experiments, and I'm not aware of any work which was have done during neurodevelopment on in early life stages. So, are there key symptoms or disease stages that animal models consistently fail to replicate? Yes, for example, tremor. Tremor, we can't have it in rodent models. So, and it's a major symptom in human Parkinson. So it's not always easy to have all important or key symptoms in an animal model. But since we are sure that the nigrostriatal dopamine circuit is impaired, so we are mimicking the human disease. So I hope it answered your question. What are the biggest limitations of current animal models in environmental Parkinson's research? Validation. Validation, because in the literature, most labs work with punctual induced PD in animals, either with using LPS or 6-OH dopamine. Um, these are the mainly used models. Uh, I believe that these models are not, I can't say not good, but not optimal, because you induce a neurodegeneration in a few days uh, um, interval, and it doesn't mimic the progressive characteristic of the human disease. In human, the disease take many years, 15 or 20 years of progression. So we need environmental Parkinson disease animal model that mimic this progressive, the slow progression, so we can see what happens into the brain during this time frame or during this progression. The biggest limitation now is to have a good model to validate a good model of environmental or toxic PD. Do your animal models incorporate factors like aging, diet, or stress that might modify environmental risk? Not for now, but it's on my perspective. So I'm working. My focus for right now is to validate a toxicant model, to have a model of a toxicant, and I guess I have one now, which is the paraquat and manip, because I tried with lead first, then tried with manganese, and now we have more good results with paraquat and manip or pesticide, and I think I can push it more to validate this model and try it with different ages or different situations. But can current animal models replicate the long prodromal phase seen in Parkinson's disease? I don't understand very well your question. Current animal models, as I said, most animal models that we can find in literature are models that induce the dopamine loss in a frame time of a few days. So you inject intracerebrally 6 hydroxydopamine or H dopamine, and in few days you have a comprehensive loss of dopamine fibers in the striatum. So in my opinion, this model are not representative of the uh, progressive character of Parkinson's disease. We also have rotenone and pesticide, and I think if we try to develop research more in these toxicant models, we can come out with a better model than LPS or 6-OH dopamine. That's my conviction, my point. Okay. Okay, thank you so much. Can't hear you again. I'm sorry.
Professor Neza?
Yes, now I can hear you.
I hear me now. Thanks so much
Yes.
for the wonderful. On behalf of Install Africa and the Flight Institute of Research and Translation, FIRAT, I would like to express my sincere gratitude to everyone who made this international webinar possible. To our distinguished speaker in person of Associate Professor Neze Abuado, thank you for sharing your invaluable insight on the topic. Your global perspective has truly enriched this session. To our attendees, we see you joining from different countries. Thank you for your patience, your engagement, and your thoughtful questions in the chat. We know many of you joined very early, while some they joined late, but your commitment means the world to us. A special thanks goes to the backend staff in person of Malam Abdulaziz, who ensured a smooth transition, stable connection, and accessibility for everyone. So to the I would like to say is you stay connected with all our social media platforms where we shall inform you on our next session. Thank you all once again, and we look forward to seeing you at future events. Thanks so much. But before you go, please kindly watch out for our STEM Talk series on what we merge together science, technology, and mathematics. What is the innovation behind it? And likewise, we also have a health dialogue series, which you need to keep looking for. Thanks so much, everybody, for the wonderful opportunity. I appreciate everybody be here. Thanks so much.
Thank you. Thank you so much.